Now, before we get into food allergies and food sensitivities, we need to understand oral tolerance. That's when everything's working correctly, and that is our goal. It means that our immune system learns to ignore things that we eat or harmless germs in our gut so it doesn't attack them. This helps stop allergies and unnecessary inflammation. When food or friendly bacteria enter our gut, special immune cells tell the body to stay calm and not react strongly. This happens mainly in parts of the gut called Peyer's patches and lymph nodes in GALT, the gut-associated lymphoid tissue. The immune system creates peacekeeper cells called regulatory T cells that keep other immune cells from overreacting, and this way the body only fights real dangers like harmful germs, not everyday harmless things. If oral tolerance doesn't work well, we can get food allergies or gut problems. So oral tolerance is very important for keeping our immune system balanced and healthy. So what types of antigens contribute to oral tolerance? Oral tolerance is primarily established through the gut immune system exposure to a wide range of antigens entering our gastrointestinal tract, and those include our dietary proteins, harmless environmental antigens, and our commensal bacterial flora. These harmless antigens are regularly encountered in the gut and must be tolerated by the immune system to prevent harmful immune responses. The mucosal surface of the gut constantly encounters pathogens and harmless antigens, and its local immune system discerns between the two, promoting tolerance towards the latter to maintain homeostasis. Specialized antigen-presenting cells, like the dendritic cells, as well as specific anatomical structures, play pivotal roles in processing these antigens and initiating tolerance mechanisms. So what about the role of GALT? Our Peyer's patches and mesenteric lymph nodes, lamina propria lymphocytes, and intraepithelial lymphocytes represent the principal anatomical sites for antigen sampling and initiation of oral tolerance. Uptake and processing of antigens by GALT is essential. Through animal studies, they discovered that if you do not have Peyer's patches anymore, the presence, as you know, that can happen as we age, we lose them, the presence of mesenteric lymph nodes can in part compensate for their absence. As for involvement of immune cell populations, the establishment and maintenance of oral tolerance is governed primarily by our regulatory immune cells. And here's a list of them here. Primarily CD4, CD5, FOXP3, regulatory T cells. Let's just call them Tregs. These cells act as immune peacekeepers. They actively suppress unnecessary immune responses to dietary antigens and our commensal or friendly gut microbes, preventing inappropriate inflammation and autoimmunity. Now, the differentiation of our Tregs is influenced by signals from our dendritic cells, especially those producing retinoic acid in the gut. And we'll talk about that in a minute. Dendritic cells sample antigens in the gut, and under tolerogenic, that means tolerating conditions, they promote Treg development, and they home them into our intestinal tissue. Additionally, other immune populations, like our B cells, also contribute to cytokine milieu necessary for tolerance, specifically by supporting low-dose antigen-induced regulatory circuits. And of course, our gut microbiota exerts profound effects on the development and function of mucosal immune cells that mediate oral tolerance. Species like Clostridia, for example, promote the generation and functional activity of Tregs through the production of metabolites like short-chain fatty acids. So a healthy and diverse microbiota is important for both inducing and maintaining the regulatory immune environment necessary for oral tolerance. Dysbiosis can compromise the integrity of tolerance and predispose us to immune-mediated diseases. What about the influence of antigen dose and frequency? So antigen dosage and the frequency of exposure are critical factors shaping the nature of oral tolerance. Low-dose antigen exposures preferentially stimulate the development of Tregs that secrete immunosuppressive cytokines, like interleukin-10, TGF-beta, and interleukin-4, fostering active suppression of immune responses. But in contrast, high doses of antigens tend to induce tolerance also, but through cell death or functional silencing of reactive T cells. When there's continuous high-dose exposure, that's going to typically favor interleukin-10 induction, enhancing tolerance, while intermittent lower doses more prominently induce interleukin-4, reflecting distinct regulatory pathways involved in immune modulation. The frequency of antigen exposure is influential. While single or repeated low-dose exposure can establish tolerance, excessive and continuous exposure could eventually tip the balance towards immune reactions rather than tolerance, especially with microbial antigens. Of course, genetics and epigenetics can play a role. Now let's move on to age and developmental stage. So the capacity to establish oral tolerance develops along with the maturation of the gut immune system, which commences prenatally and continues postnatally. Early life events, such as exposure to dietary antigens, breast milk, and establishment of the gut microbiota, are critical for the development of GALT and immune regulatory populations. It's now fairly well established that it's important to give developing infants a diverse diet once they get off breast milk. The efficacy of oral tolerance can be influenced by age, with young people typically exhibiting heightened plasticity and responsiveness of immune regulation in the gut. Aged individuals, although still capable of achieving oral tolerance, may display reduced antibody responses and diminished dendritic cell function, potentially leading to differences in the strength or type of tolerance established. The window of developmental immune flexibility may thus determine our susceptibility to immune-mediated diseases later in life. More on all of this when we get into food allergies and food sensitivities. What about dietary and nutritional influences? Of course, it's very important. Adequate intake of dietary proteins is important for immune system maturation and tolerance induction. Protein-free diets reduce both the efficacy of oral tolerance and the magnitude of immune responses to allergens. Specific nutrient deficiencies such as zinc disrupt cytokine expression and Treg induction, hindering antigen-specific clonal deletion and facilitating mucosal inflammation. The timing and form of dietary allergen exposure, especially during infancy, is also vital for promoting lifelong oral tolerance and preventing food injuries. And then, of course, we have to take a look at infections and environmental exposures. So infections and local inflammation at the time of oral antigen exposure can disturb the immune milieu, antagonize Treg functions, and favor pro-inflammatory immune activation. Of course, that's going to impede oral tolerance. And then environmental pollutants, such as components of diesel exhaust particles, can selectively inhibit the establishment of certain types of oral tolerance by interfering with regulatory pathways, including both Th1 and Th2 responses. Remember, those are types of T helper cells. The breakdown of oral tolerance in the context of persistent infection or chronic inflammation is considered an important trigger of immune-mediated pathology. And then if you want to come to the database, you can take a look at this summary table of all these different factors that influence oral tolerance. But for now, I just want to come back up to the top because there are some related topics that are worth exploring. Remember, we've talked a lot about vitamin A and how it's the most important vitamin for the gut. So we're going to discover how important it is in terms of developing oral tolerance. Okay, so let's start with retinoids, or vitamin A. So the term vitamin A refers to retinol, or all-trans retinol, and a family of biologically active retinoids that are derived from these. They're only found in animal tissues, where they're essential to innumerable biochemical processes. However, their biosynthetic precursors are plant carotenoids, also called provitamin A, of which beta-carotene is the most efficient and occurs in green parts of plants in seed oils. In the human diet, plant sources tend to be less important than those from dairy products, meat, fish oils, and butter. So there are a number of processes that these have to go through before they're used by the body. And this section describes how they are synthesized and hydrolyzed, etc., before being used. Down here we see vitamin A deficiency in children and adult patients is usually accompanied by impairment of the immune system, leading to a greater susceptibility to infection and increased mortality rate. Let's go now to retinol. And we're just going to focus on its role in the immune system. It is a form of vitamin A. And in terms of the immune function, it enhances immune responses by promoting the production and function of white blood cells, which of course are crucial for fighting infection. Retinol is absorbed in the small intestine and transported to the liver, where it's stored as retinol esters. When needed, retinol is released into the bloodstream and transported to target tissues. In cells, retinol is oxidized to retinol and then to retinoic acid. Retinoic acid is the active form that regulates gene expression. Deficiency in retinol can lead to night blindness, increased risk of infections, skin issues, and unfortunately, excessive intake can cause toxicity. So you have to be careful with supplements. As far as its benefits in skincare, that's here. There's a lot about using it for skincare. And there, of course, are some considerations and precautions. Now let's move to retinoic acid. Retinoic acid as an immune balancer. It's a molecule made from vitamin A that plays a key role in keeping our immune system balanced, especially by acting on cells that handle inflammation and protection in the body. It acts much like a manager. encouraging the body to avoid damaging excess inflammation while keeping up defenses against invaders, such as bacteria and viruses. Lowering pro-inflammatory signals. One way retinoic acid works is by slowing down the production of pro-inflammatory signals, which are chemical messages that cause the body to create swelling and redness as part of the immune response. For example, retinoic acid reduces levels of cytokines like tumor necrosis factor, interleukin 1b, and interleukin 6 in immune cells called monocytes and macrophages. When these cells are exposed to something that would normally set off a strong response, like bacterial parts called lipopolysaccharides. By lowering these pro-inflammatory cytokines, retinoic acid helps prevent too much inflammation that could damage our tissues. It boosts anti-inflammatory responses. It not only slows down inflammation, but boosts the production of anti-inflammatory messengers. These molecules help calm the immune system and promote healing after the body's dealt with an infection or injury. It encourages the growth and function of special immune cells, like the Tregs, which act as the peacekeepers. It helps regulatory T cells grow. A key role of retinoic acid is to promote the development of Tregs, vital for telling the immune system when to stand down and not attack harmless things like food or healthy body tissues. Retinoic acid, made by certain gut cells and gut dendritic cells, works together with another signal called TGFB to turn naive T cells into Tregs, increasing their numbers and ability to calm the immune system. It acts on dendritic cells. It plays a major role in shaping dendritic cells, which are the body's patrol cells that help show immune attackers what to fight. When dendritic cells are treated with retinoic acid, they become more likely to support development of anti-inflammatory Tregs, rather than pro-inflammatory cells that could worsen autoimmune or allergic problems. This effect helps the immune system react less to harmless things like food and more to real threats like viruses. They also directly block inflammatory pathways. At a deeper level, retinoic acid can block major inflammation pathways inside cells, like NF-kappa beta. Its pathway usually turns on many genes that lead to inflammation. So when retinoic acid blocks it, there's less production of the molecules that drive swelling and tissue damage. As a result, retinoic acid is able to stop inflammation, not just at the surface, but at the root, at the gene level. It modifies cell function in the gut and beyond. In the intestines, retinoic acid helps shape the immune environment to be more tolerant and less likely to overreact to food proteins or helpful gut bacteria. If there isn't enough retinoic acid, the balance shifts and the immune system can become prone to unnecessary inflammation, as seen in diseases like inflammatory bowel disease. Supplementing retinoic acid can restore balance, reduce disease symptoms, and strengthen the peacekeeper Treg cells. It's important to note that the effects of retinoic acid depend on the body's overall environment. If there are strong inflammatory signals already present, like high interleukin 15, retinoic acid may not fully prevent inflammation and can sometimes even increase it in certain scenarios. The outcome is going to depend on what other signals are present along with it. And here's a summary. And then, of course, we've got a bunch of studies. So let's take a look at autoimmune or inflammatory diseases. The central role of retinoic acid in gut immune regulation. We've already seen how it plays a direct influence on differentiation and function of Tregs and orchestration of mucosal tolerance in response to dietary antigens and microbiota. In the gut, specialized dendritic cells, particularly those expressing the CD103 marker, enzyme catalase, the synthesis of retinoic acid from vitamin A, underpinning a critical control point in immune regulation. The availability of retinoic acid is essential to drive the differentiation of naive T cells into FOXP3 Tregs, which suppress aberrant immune activation and maintain tolerance to harmless antigens present in foods. So what is the mechanism of retinoic acid synthesis in the gut? Production of retinoic acid in the gut is tightly regulated. It's a multi-step process executed mainly by gut-associated dendritic cells. The key enzymes involved include retinol dehydrogenase and most critically retinaldehyde dehydrogenase, RALDH, which converts an intermediate of vitamin A into retinoic acid. Regulation of these enzymes, particularly within these CD103 dendritic cells in the intestinal lamina propria and the mesenteric lymph nodes, forms a biological foundation for healthy immune regulation in the digestive tract. Effects of disrupted retinoic acid synthesis when it's impaired due to vitamin A deficiency, dysregulation of enzymatic machinery, or inhibitory factors, such as prostaglandin E2. The intestinal immune environment's fundamentally altered, specifically the loss or reduction of retinoic acid production leads to a diminished capacity of dendritic cells to induce FOX3 regulatory T cells, critically undermining the suppressive arm of the gut's immune network and tipping the balance towards activation of pro-inflammatory T cells. In a nutshell, if production of retinoic acid is somehow compromised, we have a loss of oral tolerance. So this leads us back to the gut microbiome. In this section, retinoic acid and microbial dysbiosis. Dysbiosis influences retinoic acid synthesis and immune regulation. Microbial dysbiosis, an imbalance in the composition and functional diversity of the gut microbiota, perturbs the whole biochemical environment that's necessary for optimal retinoic acid synthesis in the gut mucosa. A balanced microbiota provides crucial signals, often through microbial-derived molecules and engagement of those pattern recognition receptors, such as toll-like receptors, that maintain and enhance the ability of dendritic cells to express the necessary enzymes to carry out retinoic acid synthesis. Of course, the consequences, well, we know retinoic acid is the central driver of the differentiation of Tregs, especially the ones that play a critical role in establishing and maintaining oral and mucosal immune tolerance. As synthesis of retinoic acid falters, the immunological landscape of the gut quickly shifts toward a pro-inflammatory state. Restoration of retinoic acid levels in these contexts can revert dendritic cells back to their tolerogenic phenotype, reinvigorate Treg induction, and suppress disease progression, a demonstration of the molecule's regulatory dominance. So there's feedback between microbial dysbiosis and immune dysregulation. The interplay is inherently bidirectional. A disruption in microbial communities impedes retinoic acid production and Treg generation. The weakening of immune tolerance further amplifies dysbiosis by allowing inflammatory processes that damage the epithelial barrier and alter the gut's molecular milieu. And this fosters a self-reinforcing cycle of microbial imbalance and immune dysfunction, which can be observed in various forms of chronic inflammatory and autoimmune gut diseases, including irritable bowel disease and even multiple sclerosis. Importantly, evidence indicates that interventions aimed at reconstituting healthy microbiota improves mucosal retinoic acid synthesis and restores immune homeostasis, supporting this cyclical hypothesis. So there's more in this section that you guys can go through. But we notice here that restoring the microbial environment can help restore retinoic acid. So it's not just taking vitamin A, it's also having a healthy gut microbiome. You'll see here probiotic strategies for retinoic acid. Again, interplay between probiotics, retinoic acid, and gut immune regulation is a promising field for therapeutic interventions aimed at enhancing immune tolerance, maintaining mucosal integrity, and preventing immune-mediated disorders. And I'm not going to go through all of this, you can read it yourselves, but it covers mechanisms linking probiotics to retinoic acid synthesis, about probiotic modulation of the gut microbiota. And we see here, for example, increasing populations of beneficial bacteria such as Bifidobacterium and the Lactobacillus species facilitate production of short-chain fatty acids and other metabolites that support dendritic cell activity. Probiotic strains like Bifidobacterium thermophilum and symbiotic interventions, that means probiotics plus prebiotics like fructooligosaccharides, have demonstrated increased levels of beneficial microbes, improved metabolic outputs, and enhancement of microenvironmental factors favorable to retinoic acid synthesis. Similarly, interventions with fungal probiotics have boosted the abundance of acetate and butyrate producing microbiota associated with better mucosal health. In this section, you can read about how probiotics impact dendritic cell function and immune tolerance. This section will explain synergistic interactions and molecular pathways. Let's scoot down further to this section, probiotic interventions supporting retinoic acid-mediated Treg differentiation. So evidence from various studies showed that probiotics can increase the number and function of Tregs in the gut by supporting retinoic acid synthesis. In particular, Lactobacillus casei has demonstrated an ability to modulate immune responses and decrease inflammatory gene expression in the gut, facilitating retinoic acid-dependent Treg induction and suppressing gut inflammation. If we scroll down further, you'll see a summary table here about the strategy, the mechanism, the effect on retinoic acid synthesis and immune regulation, and representative findings. So, modulation of gut microbiota with probiotics increases populations of Bifidobacterium and Lactobacillus supporting retinoic acid synthesis, and that increased short-chain fatty acid. Symbiotic interventions are good. That's always recommended. That's where you combine prebiotics with probiotics, and because that's going to enhance the beneficial bacterial survival and metabolic activity. You'll get better colonization. So now let's move on to another very important factor for maintaining and creating oral tolerance, which is going to be prostaglandin E2. That is a bioactive lipid mediator produced in mucosal tissues, including the gut. It's produced through the action of cyclooxygenase enzymes If you've ever seen COX-1 or COX-2 inhibitor, COX-1 or COX-2, that's cyclooxygenase. Those enzymes act on something called arachidonic acid in response to various stimuli like tissue stress, immune cell activation, various microbial signals. And prostaglandin E2 is a key modulator of the immune system with both pro-inflammatory and immunosuppressive properties relative to the delicate balance required for oral tolerance. And in this section, you can learn more about how it works, but I'm just going to cut to the chase and tell you that this is something that can be seriously disrupted by the use of non-steroidal anti-inflammatory drugs, NSAIDs, like Tylenol, for example. And so, coming back to oral tolerance, two very important factors are having enough retinoic acid, which comes from, it's a metabolite of vitamin A, and functioning prostaglandin E2 that isn't being chronically interfered with by the use of non-steroidal anti-inflammatory drugs, NSAIDs. So let's move on to what medicinal herbs might help. There is some evidence from preclinical and some mechanistic studies showing that certain herbs or herbal formulations can influence the cellular and molecular pathways that underlie oral tolerance, though the effects can be contact dependent and of course vary on the specific herb dose and mode of administration. So turmeric, curcumin, as a potent anti-inflammatory and immunomodulatory agent, promotes oral tolerance by enhancing regulatory T-cell differentiation, suppressing pro-inflammatory T helper cell 17 and Th2 responses, and reducing gut inflammation. It supports gut barrier integrity, crucial for preventing inappropriate immune activation. And there are studies that show it reduces inflammation in irritable bowel disease and modulates immune responses in animal models of food allergy, partly by promoting the Treg cells and inhibiting NF-kappa B signaling. The use: take turmeric powder 1 to 2 grams a day in food, capsules, or tea with black pepper to enhance absorption. Be careful, high doses can irritate the gut in some individuals. Slippery elm also helps, but we try to avoid the use of slippery elm these days just because it's endangered, so I'm going to skip that part. But licorice, and that's another one of our tiles. It contains glycyrrhizin and flavonoids with anti-inflammatory and immunomodulatory effects. It supports oral tolerance by reducing gut inflammation, enhancing mucosal repair, and modulating immune responses. Deglycerized licorice is often used to avoid side effects like hypertension. As far as the evidence goes, it's been used in traditional medicine for ulcers and irritable bowel disease, with studies showing it reduces pro-inflammatory cytokines and supports gut healing, which may aid tolerance. Mechanisms for oral tolerance: well, it reduces gut inflammation, promotes balanced immune response, potentially supporting Treg activity, and preventing excessive Th2 responses to antigens. Marshmallow, and this can be used instead of slippery elm. Like slippery elm, marshmallow root's mucilage coats and soothes the gut lining, reducing inflammation, supporting barrier function. It helps maintain an environment conducive to oral tolerance by minimizing gut permeability and irritation. It's been traditionally used for gut inflammation. It's effective at soothing conditions like gastritis or leaky gut, which can impair oral tolerance. It enhances gut barrier integrity, reducing antigen leakage that could trigger immune responses. And it supports a balanced microbiome. And then astragalus, an adaptogenic herb with immunomodulatory properties, promoting a balanced immune response. It enhances Treg cell activity, reduces excessive Th1, Th2 polarization. We'll talk about that in a future class, by the way, which supports oral tolerance. It strengthens gut barrier and supports beneficial gut bacteria. Studies suggest that astragalus polysaccharides modulate immune responses in autoimmune and allergic conditions, with potential benefits for gut immune regulation. And then there's some experimental evidence here you guys can read through. And then there's a whole section down here. In the interest of time, I'm not going to read all this. I just want you to see where it is. Mechanistic insights, gut-associated lymphoid tissue, and herbal polysaccharides. So some of the herbal polysaccharides are also very useful. Polysaccharides interacting with Peyer's patches. We went over this in class. Polysaccharides derived from medicinal herbs are increasingly recognized for their immunomodulatory effects, particularly through interactions with Peyer's patches. Certain polysaccharides interact with M cells, dendritic cells, B cells, and T cells in Peyer's patches to modulate immune function, often by enhancing mucosal immunity and promoting immunoglobulin A production and or regulating cytokine secretion. An astragalus polysaccharide is described here, how it works, and its relevance to oral tolerance. Bupleurum polysaccharides, Donquai polysaccharides, Ganoderma, or that's reishi, and in general beta-glucans from various medicinal mushrooms. Here are the mechanisms of interaction and the relevance to oral tolerance. So I'm going to leave it to you guys to go through this section so that we can wrap this video up. And next class is going to be on food allergies.